Okay, so we were talking about formed elements. We'll talk about leukocytes next. Leukocytes are the white blood cells, which makes up most of the rest of the formed elements of blood. They're very important because they help fight infection, and they are the only complete cell of the blood. They have nucleus, and they have multiple organelles within inside them. Okay, there are 5 different types of white blood cells, which we will go over in very much detail. And then you have platelets, which are fragments of cells that are there specifically for clotting the blood and preventing further blood loss. Okay, most formed elements will survive in the bloodstream for only a few days. Sorry about that. Okay, so here we look at a blood smear where we're seeing multiple elements on the left, and then we have them on the right highlighted for the different types. You can see on the left we have general formation of blood, and we can see that these right here, the large one in the middle with the little lobules in it, are which type of cell. Those would be the neutrophils right there. Okay. Alright, then let's go ahead and clear that off. And then we'll look at the next different type here. We're going to find some red blood cells. Red blood cells are very easy to find. They are the pink almost, and some of them have a white dot in the center because they do not have a nucleus or they are anucleated. Those are erythrocytes, also known as red blood cells. Okay. Realized I was not doing any good doing that. Okay. Sorry about that. Okay, and down here at the bottom we have the platelets, which are just the little chunks of things in between in the blood, because they're basically parts of cells. Okay, here we look at the formed elements of the blood. We have the red blood cells. They are biconcave, meaning that they are flat in the middle with raised sides. They're kind of like a little button. Okay, then you have your white blood cells, which are larger and have a very misshapen nucleus for the most part. Okay, you've got your granulocytes, which are made up of 3 different types. You have your neutrophils, basophils, and eosinophils. What's interesting about the eosinophils, it has a red color like if it were a red blood cell. Okay, they're usually stained red or bright red, and they release chemicals that help reduce inflammation. Okay, then you have your agranulocytes, such as your lymphocyte and your monocyte, and finally you have your platelets. Alright, I'm going to go ahead and put our first question here. Alright. Question number 1. This blood cell is of what type of blood cell? Question number 1. Okay. Very simple. So back. Alright, production of formed elements. Got a little video for this one. Alright. We'll check that out very first and foremost.
At the Watershed, we believe in recovery.
Yeah.
We believe that every alcoholic and addict deserves.
Here we go.
Amanda Hasudugan, biology and medicine videos. Please make sure to subscribe, join the forum group for the latest videos. Please visit Facebook at Amanda Hasudugan. In this video, we're going to look at the red blood cell life cycle, or erythrocyte life cycle. So here I'm drawing a blood vessel. The blood vessel contains red blood cells. here are mature red blood cells, and mature red blood cells are responsible for transporting oxygen around our body. But where do the red blood cells come from? Well, they all arise from the bone marrow. So here I'm drawing a bone. The red blood cells come from stem cells within the bone marrow. Erythropoiesis is the term used to describe erythrocyte synthesis, or the production of red blood cells. Hemopoietic stem cells can differentiate into pro-erythroblast and then early erythroblast. In the early erythroblast stage, we see ribosome synthesis, and this is important because once the early erythroblast becomes a late erythroblast, the ribosomes are synthesizing a lot of hemoglobins, and so we have hemoglobin accumulation. Hemoglobin is the molecule within red blood cells that actually carry the oxygen. So this late erythroblast can then develop into a normoblast, and normoblast already contains all this hemoglobin. A normoblast will then become a reticulocyte. A reticulocyte loses its nucleus. However, it is not a red blood cell just yet. A reticulocyte actually stays in the bone marrow for several days before entering circulation. And once entered into circulation, the reticulocyte, after about 1 to 2 days, or 24 to 48 hours, it will mature and become a erythrocyte. Erythrocytes are the red blood cells that we know that circulate and carry oxygen around our body. But erythrocytes don't circulate in our body forever. It has a lifespan of about 128 days when it's aged or when it's damaged. And so when it's aged or damaged, it has to be removed. Red blood cell removal occurs in 3 main organs: the spleen, the liver, and the bone marrow. So here I'm drawing a few old red blood cells that need to be cleared up. And here I'm drawing the spleen. Within the spleen we have macrophages. And it is these macrophages that will actually engulf these old erythrocytes and break them down. And so when the old red blood cells are broken down, you end up with a lot of hemoglobin. The hemoglobin can then be broken down further into globin and its heme component. Globin is then broken down to amino acids, and the heme is broken down to 2 main products: bilirubin, essentially, and iron. These macrophages are not only found in the spleen, as I mentioned, but they're also found in the liver and bone, and they do exactly the same thing. Most of the breakdown product of red blood cells are recycled. So for example, here the amino acids will enter back to circulation and then will travel to the bone marrow to be used in erythropoiesis again, the production of red blood cells. The iron and bilirubin also have similar fates. So the iron, it can't actually travel in the blood just like that. It has to be attached to a transporter. And so iron obtains a iron transporter from the liver known as transferrin. And it's this way how iron makes its way back to the bone marrow for erythropoiesis. Bilirubin goes back to the liver, either through albumin transporter or whatnot. But essentially, bilirubin ends up in the liver. Now, bilirubin has a few fates. Most of the time, bilirubin is actually secreted or excreted through the bile system. So bilirubin will enter the bile and then will be secreted into the duodenum of the small intestine. And this is through the bile duct. So bilirubin is excreted in feces, or it is reabsorbed and is excreted in urine. Now that we're in this picture of the stomach and small intestine, it's important to note that when we eat nutrients, some of these nutrients are important in red blood cell production. They are absorbed in the small intestine. So nutrients absorbed for erythropoiesis include amino acids, monosaccharides, lipids, vitamin B12, folic acid, and iron. And all these are used in erythropoiesis because we need these to make red blood cells. And as I mentioned, the main function of red blood cell is to transport oxygen to tissues. So now let's look at one important hormone or factor that influences erythropoiesis. In times of hypoxia, which when we have a decrease in oxygen levels, this will stimulate the kidneys to produce an important hormone called erythropoietin. And erythropoietin is such an important hormone in erythropoiesis because it stimulates erythropoiesis. And so when you have kidney failure, for example, your kidneys are not able to produce erythropoietin, so you usually develop anemia. I hope you enjoyed this brief video on red blood cell life cycle. Thank you.
Okay, so most blood cells do not divide, but are renewed by stem cells. Let me check on something really quick. I apologize for doing this. Okay, I'm just making sure I wasn't muted. I didn't know. Okay, hematopoiesis is the process of blood cell reproduction, or blood cell production, excuse me. Okay, and what happens is it occurs in a couple different places. It happens in the fetus. It would happen in the liver, the thymus, the spleen, lymph nodes, and in the red bone marrow. Okay. But after birth, when you become an adult, and even as a small child, okay, it will happen in the red bone marrow. Okay, the red bone marrow in either the axial skeleton and girdles, or in the epiphyses of the long bones, the humerus, and the femur. Some white blood cells are produced in lymphatic tissue, but most of those are produced in the red bone marrow. Hemocytoblasts give rise to all of the formed elements of blood. Okay, and growth factors will determine the type of formed element that will be derived from that stem cell. So here's a look at the hematopoiesis. Okay, the progression of hematopoiesis starts off with several different progenitor cells. Okay, those early on in the process. Okay, you have the proerythroblast. Okay, and those proerythroblast ends up being a red blood cell. You can see the proerythroblast and how things will change. So as it goes from proerythroblast to early erythroblast to intermediate erythroblast, they have a nucleus in the center of all three. Okay, the late erythroblast has a nucleus that is basically no longer needed, so it is ejected, or what's called extruded. Okay. After that, you have a reticulocyte, and that reticulocyte will then become your mature red blood cell. All right. Myoblast. Myoblast will basically make up three different types of white blood cells eventually. The myoblast will differentiate into a progranulocyte. Okay, the progranulocyte will make up the three different granulocytes eventually, depending on the signals from the body on which of those end up getting produced. Okay, basophilic myelocytes, eosinophilic myelocytes, and neutrophilic myelocytes will neutrophilic myocytes will then be the next stem cell in the production of mature granulocyte cells. So here we see them going through the process, and eventually they end up being the mature granulocytes, the basophil, eosinophil, and the neutrophil cells here. Lymphoblast, it's a one-step process. Lymphoblasts go directly to lymphocytes, and monoblasts go directly to monocytes. Okay, and over here we get a little bit of a crazy looking process here. We have a megakaryoblast. Okay, the megakaryoblast will differentiate into a megakaryocyte. The megakaryocyte will then break up into many different pieces, and it will form the platelets. Get my computer back to control. Okay. Red blood cells. Red blood cells are biconcave discs that are anucleated, meaning they do not have a nucleus, and they have essentially no organelles. The red blood cells are dedicated to the respiratory gas transport, which means it's moving oxygen, carbon dioxide, and it does this by using hemoglobin. Hemoglobin is a protein that functions in gas transport. What makes it unique as a protein is it has a heme molecule, and that heme molecule contains iron, which allows it to anneal to that oxygen and hold on to it. Okay, the red blood cells are an example of how structure will fit the function. Okay, without that structure, it would not have the function that it does. The biconcave shape has a huge surface area. Okay, it has a huge surface area relative to the bottom, or the volume, excuse me. So here, if we have basically like a red blood cell like this, you have a lot of surface area on the top, a lot of surface area on the bottom. But however, if I open my hands like this, I've got the same amount of surface area as I had before, but I've got a lot more volume. Okay, you can see it better that way maybe. Here, a lot of surface area, not a lot of volume because there's barely even a hole there. Here I open it up, same amount of surface area, but not near as much volume, if you understand. Okay, the biconcave shape allows the red blood cells to bend or fold around their center. It gives erythrocytes their flexibility and allows them to change shape as necessary. So they're constantly having to change shape to fit through those little tiny openings to get into cells, because they've got to fit between other cells in order to get to the tissues. More specifically, those areas such as the capillary beds, and then entering into tissues from those capillary beds. So here we look at some erythrocytes. Okay, we have the erythrocytes under extremely high magnification here on the left. Okay, you can see these little dots and indentions on it. What might those be? What might those things be on those erythrocytes? That's question number 2. All right. So those erythrocytes we looked at over here, and we can see the erythrocytes in the tissues. See the erythrocytes in the tissue here? What kind of tissue might this be? Okay, it's probably a capillary bed full of red blood cells. Okay, erythrocytes. More views of them. Here we are looking at them within the capillary beds. We see the erythrocytes are highlighted in more of a blue turquoise color at the bottom left, and then you see the nuclei of the endothelial cells on the right. Okay, it must pass between these cells in the capillaries in order to make it out to the tissues. So they have to be able to change shape, as mentioned before, in order for that cell to leave those capillary beds and then go to the tissues. Okay. Oh, excuse me. Alright, so here we are looking at more erythrocytes. See how the erythrocytes look really odd when you look at them from the side. You're used to looking at them from the top, and they kind of look like a cup. Well, if you look at them from the side in a cross section, they're really fat on the ends and really skinny on the middle, which makes them very easy to bend. Okay, if you don't have much membrane in the middle to bend, then you have a very easy time fitting through those tight areas in which need to be accessed by these red blood cells. Okay, so let's look at the red blood cells here under high magnification under scanning electron microscopy. How do we know it's scanning electron microscopy? It says it right there, SEM. Okay, and here we're looking at the red blood cells, the white blood cells, and then the platelets. And in comparison, you can see there's quite a bit of difference. Okay, under scanning electron microscopy, we see the platelets just look like a bunch of little junk floating around. Okay, it's by as good a description as you can get. Little pink pieces of junk. They match the color of the red blood cells pretty well, but that's about all you can say that they have in common. Okay, the only functioning cell, complete cell, that has a nucleus and has organelles that function and carry out different processes would be the white blood cells. And then you have what we've been talking about, the red blood cells right here. And the red blood cells are what we're most interested in at the moment, talking about oxygen and how it's carried throughout the body. Alright, let me get control back. There we go. Alright, so as I told you last time, I can't show these videos, but you know it's in the PowerPoint presentation. I really heavily suggest you watch these, because it's pretty good in its description of what's going on in the cell. Alright, more about red blood cells. And I've talked about hemoglobin just a little bit. We're going to hit a lot more on hemoglobin now. It accounts for about 1/3 of the cell's volume. 1/3 of the red blood cell's volume, is hemoglobin. It consists of these things. Okay, it consists of a protein globulin. Okay, it's made up of 2 alphas and 2 beta chains, and each of those chains is bound to a heme group. Okay, well, what's a heme group? Well, each heme group bears an atom of iron, which I said iron is very important to the blood, and this is why. Because without the iron, it wouldn't be able to bond or bind the oxygen. So iron is very important to your diet, and that's why you see a lot of problems with people that are anemic and have a low iron content, and they have to take extra iron in order to build a stronger amount of red blood cells in their body. Heme molecules are transporting oxygen, okay, and iron is a requirement in order for that to happen. The oxygen content determines the blood color. The more oxygenated it is, the brighter red it is. The most deoxygenated blood is extremely dark red. That means that patient, if you see really dark red blood, is not getting very much oxygen to the tissue. Okay, globin molecules will then transport carbon dioxide as well. Okay, the carbon dioxide, remember, it's on its way out for the most part, but it does play several important roles while it's in the body as well. Okay, and one red blood cell contains 250 million hemoglobin groups. One red blood cell contains 250 million hemoglobin groups. Thus, it can carry 1 billion molecules of oxygen. That's one red blood cell. Question 3. Okay. How much oxygen, how many molecules of oxygen could, sorry, I don't know what just happened, I have no idea what's going on. Okay. Could 100 red blood cells carry? How much oxygen could 100 red blood cells carry? If one could carry 1 billion, how much oxygen could be carried? Alright, so here we are looking at the hemoglobin and looking at the structure of it. The globin molecule, the heme particles in the middle. Okay, see how in which the heme is actually found in the center right here. Okay, and in the center of each heme group, you have the iron. Why is the iron important? Well, here it goes back to having an understanding of chemistry. Okay, iron is basically a nice little positive charge. And when you have iron as the ion, it's a +3 or a +2. Because it can lose either 3 electrons or 2 electrons. Okay. Well, if you get iron that has enough charge that it's not really forming an ion, but it's got enough charge to attract your nonmetals, such as these nitrogens that you find in this area right here. Those nitrogens are going to be attracted to that iron. Okay, because iron is a very positive type of atom, and since nitrogen is a very negative type of atom, very electronegative, they're going to be attracted to each other. And iron thus is going to provide that same type of thing for when oxygen comes in, because the iron is even going to be even more attracted to the oxygen than it is to the nitrogen. Okay, think about in this case, the nitrogen is you're going somewhere, somebody you don't really like, and then you walk in, and then your future wife or future husband comes in, and you're like, wow. Okay, oxygen is that. Okay, oxygen is what it wants to be with, and nitrogen is what's basically just kind of taking him to the ball game. Okay. All right, red blood cells. Transport of oxygen and carbon dioxide. Oxygen that's transported bound to hemoglobin makes up 98.5% of the oxygen that is being transported. Okay, there's that 1.5% of it is attached, or not, not attached to anything, and it's just free floating in the plasma. It's almost like dissolved oxygen in water. Okay, and then you have hemoglobin. There's some hemoglobins that are just kind of lost and floating around, but that's very rare. But as I said before, transported attached to hemoglobin is 98.5%, and each hemoglobin binds 4 oxygen atoms. And remember, each red blood cell has 250 million red blood cell, hemoglobin molecules per red blood cell. 250 million, and each one can carry 4 oxygen atoms. So each cell can carry 1 billion. Each red blood cell can carry 1 billion oxygen molecules. Carbon dioxide is dissolved in plasma, okay, accounting for about 7% of it. And carbon dioxide transported as the bicarbonate ion, which is the blood buffer system, which controls the pH for the entire body for the most part, makes up 70%. So carbon dioxide has a very important role. Not just a waste product, it has a very important role in the body. And it's also found chemically bonded to hemoglobin at a rate of about 23%. The downside of that, since it has hemoglobin has a little bit of an attraction to carbon dioxide and a lot of attraction to oxygen. Well, there's carbon monoxide. You've heard of carbon monoxide, right? Carbon monoxide poisoning. Well, carbon monoxide looks a lot more like oxygen than it does carbon dioxide when it comes to hemoglobin. There's that free oxygen on one side of the carbon monoxide molecule. It's very electro-rich and very electronegative, and that gets bound to hemoglobin more rapidly than carbon dioxide would, and would cause deoxygenation of the blood and rapid decline of oxygen content. Okay, red blood cells. Transport and exchange of carbon dioxide. Carbon dioxide diffuses into the red blood cells and combines with water to form carbonic acid, which will quickly dissociate into hydrogen ions and bicarbonate ions. It's a buffer system, okay, meaning that depending on how high one is on the right, okay, if you increase the amount of bicarbonate ion, okay, we'll go the other direction and start forming more carbonic acid. If you increase the hydrogen ion concentration, it will form more carbonic acid. If you get a lot more carbonic acid building up in the blood and it increases, then you will see it going this direction into bicarbonate ion formation, or completely breaking down into carbon dioxide and water. So as this one increases, it breaks up two different directions. It goes this way and that way. Alright, if you have extra carbon dioxide buildup in the body, it will first form carbonic acid by combining with water. Well, after that builds up to a certain degree, it's going to break down into bicarbonate ions. It's a three-way buffer system. Most buffer systems that you'll learn about, especially in chemistry, are only a two-sided buffer system, where we have a three-sided buffer system here, based on the complexity of the way in which carbonic acid is held together very weakly. Okay, in red blood cells, carbonic anhydrases will reverse catalyze the conversion of carbon dioxide and water to carbonic acid. Okay, here we see the red blood cells. Erythropoiesis is the production of red blood cells. Hemocytoblast is transformed into a proerythroblast, and that proerythroblast will then develop into an early erythroblast. Then the development pathway consists of three different phases. Ribosome synthesis in the early erythroblast starts the production. Okay, hemoglobin accumulates in the intermediate erythroblast, and then in the late erythroblast, and then finally the ejection of the nucleus from the erythroblast starts the formation of reticulocytes. And reticulocytes are simply immature red blood cells. Okay, reticulocytes are released from the red bone marrow into the circulating blood, which contains about 1 to 3% reticulocytes. And then these reticulocytes, or immature red blood cells, then become mature red blood cells, or erythrocytes. Okay, circulating erythrocytes. The number remains constant and reflects a balance between red blood cell production and destruction. There are too few red blood cells, leading to hypoxia. Okay, if you have too few red blood cells, it will lead to hypoxia. Okay, if there are too many red blood cells, okay, it causes an undesirable blood viscosity, which means that it becomes more syrup-like and makes it harder to flow through the blood vessels. Okay, that's not always good, because there's people that have to take blood thinners. Well, over time, if you have too many red blood cells, mostly those that are not even functioning anymore, you get a very thick blood that's not flowing well, and it can cause major problems for the body. Erythropoiesis is hormonally controlled and depends on adequate supplies of iron, amino acids, and the B vitamins, both folic acid and B12. Okay, folic acid and folate are basically the same thing, just so you know. What the difference is folate with one more hydrogen is folic acid. Take folic acid, remove a hydrogen, it's folate. So folate's basically the salt of folic acid. Okay, erythropoietin, EPO, which is released by the kidneys, is triggered by hypoxia due to decreased red blood cells, decreased oxygen availability, as well as the increased demand for oxygen. Enhanced erythropoiesis increases the red blood cell count in circulating blood, and oxygen carrying ability of the blood is increased as well. Okay, red blood cell production. Okay, decreased blood oxygen triggers the kidney, and it releases EPO, erythropoietin. And that erythropoietin then moves to the bone marrow, and that bone marrow then starts the process of red blood cell production. And then that increases the amount of red blood cells, so this increases the amount of oxygen flowing through the body. There's a lot of different athletes that are now using EPO or erythropoietin as a performance-enhancing drug. It's one of the things that most of the cyclists got busted for as their means of increasing their production. Red blood cells. The lifespan of an erythrocyte is between 100 and 120 days. Okay, old red blood cells become rigid and fragile, and they can no longer bend and fit through the gaps that they need to get into in order to get to the tissues. And then their hemoglobin begins to degenerate. Okay, dying red blood cells are engulfed by macrophages located in the spleen or the liver. The heme and the globin are then separated as the iron is salvaged for reuse. Globin chains are broken down to individual amino acids and then are metabolized or used to build new proteins. Then the iron that's left over gets released from the heme and is transported to the red bone marrow in order to produce new and more hemoglobin. Heme then becomes bilirubin and is secreted in the bile, and in the intestines, bilirubin is converted by bacteria into other pigments. It gives the feces its brown color, and then it gives urine its yellow color. Okay, I will be posting more very, very soon. The blood cell chapter will have 2 parts, and I will also get started towards the next test, and I will try to get as far ahead as we possibly can. Pay attention to the test posting. The test posting will be occurring on Thursday. Okay, and it will keep on for about a week's period of time. I don't remember what time I set for the final closing of that. Okay, and there will be one more video for this lecture posted